Okay, good afternoon, everyone. Welcome back. I'm very glad today to chair and coordinate this session. Actually, maybe many of you who were here in this fantastic school in Urbino know that this is the second edition of this roundtable. Because how many of you were here? No one, so like me. I was not here because this is my first year as a president of the Division of Medicinal Chemistry of the Italian Chemical Society. You know myself, my name is Maria Laura Bolognesi, but for those of you who are connected on the web, I would like to introduce myself and also to tell a little bit more about the organization of this session. So I took over, as I said to you, this is the second edition. And so I took over for this responsibility from the president of last year, Gianluca Sbardella. I'm sure that he will be connected. And I said, okay, another edition. So again, COVID, maybe an update is okay, but as a scientist, I'm also a little bit worried about the COVIDization of science, okay? But then I said, no, it's very good to have a round table, and in a few minutes you will understand why I'm so glad today to be here. So this is, as I was saying before, this is an event organized by the Division of Medicinal Chemistry, and before to start, especially even for you who are in this room, I don't know if you know what is the Division of Medicinal Chemistry. So I believe that today this event is very important also to show to everyone who we are and especially what is our mission. So our mission is to improve, as you read from the slide, to improve medicinal chemistry in academia and industry by promoting contacts and collaboration among researchers working in these related fields, with aim to propose and develop projects that now I don't see because it's covered, but I believe towards medicinal chemistry. So this is truly the best environment to do that, to establish collaboration, and even to talk to experts who are not, many of us are medicinal chemists, but some of us, you will see in a moment, are not medicinal chemists, but work in areas which are strictly interrelated with medicinal chemistry. So today what we are doing is something which is in our heart, in the core mission of the division. And just some statistics about the division, which was found in 1979, as you can read from the slide. We are a quite big community. We should be proud of that because as Italian division, we belong to a larger division, which is here in the top of the slide, which is the European Federation for Medicinal Chemistry and Chemical Biology. And we had the pleasure today to have with us the president of the EFMC, who is Rui Moreira here on the left. But in any case, so this is a very special event for the division. And I would like to highlight something. Look at the member profile. So far, the member profile belongs to the university. Just but quite small amount belong to research institute and trade and industry. And when I wrote my candidacy letter to all the community, I wrote that my main aim in Italy is to put industry and academia together. And I'm quite glad that today we have several important representatives from our Italian pharma, and also we should put people together. And this is something that I trust a lot, especially in a very important and challenging field as it is drug discovery and as it is drug discovery in COVID, okay? So today, please trust me, you told me that you felt the passion in the introduction, but trust me that today I'm even more glad, more convinced, because we are putting efforts together. We have several important representatives of the Italian ecosystem, as well as from our European Federation of Medicinal Chemistry. So I believe that we will enjoy this round table, and we will learn a lot from several points of view. So I have organized this round table in this way. We will start with Professor Giovanni Maga. It's an honor to have you, Giovanni, here. This is truly an honor, because talking about COVID in Italy, you are one of the most important experts. You are not a medicinal chemist. You are a molecular virologist. This is something which is correct, or maybe you can correct. Please, Giovanni.
Absolutely correct, yes. Okay. I'm a molecular virologist.
Yes, so you are the truly expert, and you belong to an area which is very important if you want to make a drug discovery for COVID. And so that's why I'm honored and very pleased that you can give us an overview. So I would like that you start, and I also give some serious questions to all the speakers just to start the conversation. And so I would like that you can give us an overview on the situation in Italy, and especially you talk about the new variants and what we can do against the new variants and the present situation.
So thank you very much. I mean, it's me that I am honored to be part of this excellent roundtable, and thank you very much for the invitation. You know, the situation in Italy, but I would say all over Europe, is now quite unusual, because we are now experiencing a new wave of infections, which is unexpected in this period of the year. Usually summertime was sort of a relaxed, so to say, time in terms of the diffusion of the virus. Unfortunately, now we are facing a new variant, actually a family of variants, which are so infectious that even the climate factors, meaning the temperature, the social, dispersion that is typical of the summertime, humidity, the fact that we live more time open air is not enough to mitigate the diffusion of the virus. So what I will do briefly today is just to present you the identity card, the identity kit of these new variants, and to explain why they are so contagious and why they are variants of concern, especially in terms of the protection given by the vaccines that are still working. I would say they are still, a very essential, I would say, defense that we can have against the serious illness following the infection, but unfortunately they are not so efficient in protecting us from being infected. And this is, of course, a matter of the molecular characteristics of these viruses. So if I may, I will now share my screen so that I prepared, a few slides to help me illustrate the characteristic of these new subvariants of the SARS-CoV-2. Let me see. Probably I need to get the privilege because I'm not allowed to share the screen.
Just a second.
Sure.
Can you try now?
Okay, now it's working perfectly. Let me see. Magic. I hope you can see my screen now.
Yes, we can.
Okay. So we all know that we are facing a SARS-CoV-2 variant that is called Omicron. The reason is that the WHO has decided to classify those different lineages of the SARS-CoV-2 that appeared over the time of the pandemics with Greek letters, and we already have reached the Omicron, which is, let's say, midway between the whole alphabet. So the Omicron variant, the original one, is called BA.1. And as you can see, it appeared in January 2022 in South Africa. Actually, all the Omicron lineages are originated in South Africa. And one point that sometimes people didn't realize is that all the different lineages of Omicron were already present at the same time in South Africa. So they have not been, selected over time, but they were already part of the very broad genetic variability of the virus in the original countries. But then they, let's say, took over with different kinetics. So the first one was the BA.1. More or less, I would say a few months before, there was a sublineage of the original Omicron, which originated in South Africa, but it was not very present, in Europe, while the Omicron one very quickly dominated everywhere, particularly in our countries. And then we started to detect also the other lineages, in particular the BA.2 and the 4 and 5, which are those that are currently driving this new wave of infection. So one point that I wanted to make was all these variants were already circulating at the very beginning of the year, meaning that they have not been selected by vaccination or by whatever else you can think of, but they were part of the pool of genetic variability of the virus. And what we observed over the time, was that very quickly there were sub-lineages, meaning that originating from one of these variants, particularly the BA2, there were additional genetic changes that made up new subtypes of the virus, those that you can see here, which were starting to be detected very early during this year. They were spreading over multiple countries, but so far they didn't took over, meaning that they didn't replace the original Omicron and the variant number 2, while the BA4 and BA5 very quickly started, I would say starting from April, May, to become dominant and are now representing the absolute majority of the virus which is infecting people nowadays all over the world, including Europe, including Italy. So this is just to give you an idea of the broad genetic variability of this virus, which is not surprising at all, and we will see in a few minutes why we should not be surprised to observe this variability. But obviously, all these variants, they accumulated changes that in some way or another made them more efficient, and that's why they were classified as variant of concern, because most of the mutations that are characteristic of these viruses are located in the spike protein. So the problem, the issue that we are confronting now is the fact that those mutations are affecting an essential part of our immune defenses, meaning the ability of neutralizing antibodies produced either upon infection or after vaccination to engage the spike protein, which is the outer surface glycoprotein of the virus, and which is the molecular element that enables the virus to engage the AC2 receptor and then infect our cells. As I said, to affect the ability of the neutralizing antibody to block the interaction between the spike protein and the cellular reception. Why so? Well, the spike protein, you know, is also the main antigen which is used in our vaccination procedure, and it has been the subject of intense studies and molecular characterization. So we actually know basically the function of every single amino acid within the region of the spike protein, which is important for the interaction with the receptor. So this is a three-dimensional representation of the spike protein, and those parts that you see in different colors are those that are on one side important for interaction with the AC2 receptor, and on the other side they are portions that are recognized by antibodies capable of blocking such an interaction. So how can we know the function of this single amino acid? So over the months of the pandemics, several approaches have been used. But first of all, one idea is to sequence those variants that are present, you know, circulating in people, and then subject the virus to different experimental tests, putting the virus in the presence of neutralizing antibodies and see how much they can still neutralize the mutants. But we can also work, you know, from bottom up, with a bottom-up strategy, by changing, by deep mutagenesis scanning every single amino acid, and then test every single mutant against neutralizing antibodies. Or we can take the virus, make it replicate in the presence of the selective pressure of sera, for example, from vaccinated people or from purified antibodies, and see what kind of mutations accumulated that are able to reduce the ability of the antibodies to bind to the protein. So now we have really a map that enables us also to predict the effect of a certain mutation as soon as it appears within a circulating strain. And that's the reason why we were immediately able to spot what the problems were with these new Omicron variants. So we know, thanks to those approaches that I showed you before, what portion of the spike protein are most efficiently recognized by neutralizing antibodies. And you see them here in these two different views of the spike protein in the different colors. So we can distinguish all these sites that we can more properly call epitopes, meaning those portions of the protein that are recognized by our immune system. And so we can immediately look whether mutations present in circulating viruses are located within these critical regions. And that's what has been done with the Omicron variants and with the Delta variants. And it was also the reason why we started immediately to worry about Omicron. So here you can see again the three-dimensional structure of the spike protein from the Delta variant, the one that was driving the big infection wave at the end of the last year, which was dominating all over the world. And in red, you can see those amino acids that were mutated in the Delta virus, located in the region where the spike protein binds to the receptor, so which is important for engaging and infecting the cell, and also in the different epitopes that are recognized by neutralizing antibodies. Now, if we then compare this picture to the picture of the original Omicron, the Omicron 1, you can immediately realize the higher number of mutations in the critical part of the protein. And those mutations, they immediately were, you know, classified to have two main effects. On one side, they made the virus more capable of binding to the receptor. with a small difference with respect to the Delta virus, because this particular conformation of the spike protein is more capable of infecting cells from the upper respiratory tract. I won't go into too many details, but the virus can enter the cell using, let's say, two doors. One door, so one molecular mechanism, is typical of the deep respiratory tract, our lungs. The other mechanism is more efficient for the upper respiratory tract. And Omicron changed the protein in a way that made it very efficient in infecting our upper ways and less efficient in infecting our lungs, which was good. The bad news was that many other mutations were able to substantially reduce the ability of our antibodies to recognize and block the interaction between the spike protein and the cell. So the virus was more infectious and more capable of evading the immune response, either coming from previous Delta infection or from vaccination, which again, I want you to remember, is based on the spike protein of the original Wuhan strain, so the one that appeared in end of 2019, early 2020. Now, if we look at the detail of the spike protein, which is just the domain which is able to bind to the ACE2 receptor, again, by comparison, the beta variant that appeared early in 2020, and then the Delta variant that dominated in 2021, and then the Omicron, you can see all these regions in Omicron corresponds to critical epitopes that are recognized by the four main classes of neutralizing antibodies that our body produced either after vaccination or upon infection. And you see that the number of mutations are much higher than with the other variants, that we were still very sensitive to our immune response. That was the reason why the vaccination campaign was so successful at the very beginning. Now, just to give you an idea what I mean when I speak about broad genetic variability. So in these schematics that are taken from a very recent paper is representing the three main epitopes, portion recognized by neutralizing antibodies, which are located in the receptor binding domain, so the portion of the spike protein that I showed you before, which is interacting with the ACE2 receptor. I think you are familiar with the one-letter code denoting the different amino acids. So what you can see, this is the Wuhan strain, and then these are the different variants that appeared over the time, and you can immediately realize that the Omicron family is characterized by a larger number of variations in critical epitopes, meaning that these viruses are very efficient in evading the protection given by antibodies. Now, one question that very often is raised by people, okay, so if those viruses can evade the antibody response, why we still want to vaccinate people? What's for? And why vaccination actually protect us still from the serious illness? Because I just want to remember, antibodies represent one part of our immune defenses, those that are, primed to block mainly the infection. However, the second arm of our immune system, which is represented by specialized lymphocyte T cells, which are able to recognize the infected cells that are exposing pieces of viral proteins on their surface, they recognize different epitopes. So that's why the cell-mediated immunity, which is stimulated by vaccination, is still holding even against these hypermutated variants. And even though we can get infected, if we are fully vaccinated, we have much lower probabilities to develop a serious illness. But of course, we are not sure how long this protection will still, continue to protect us. That's why we need to have new vaccines that are more, tailored upon these variants. Anyhow, this was just to give you an overview of the kind of variability that those viruses have accumulated due to evolution. And I will, tell you in a moment why probably this happened in such a very short period of time. Now, the most, let's say, problematic variants that are now circulating are the Omicron BA.4 and 5. So again, this is our friend, the spike proteins, actually the receptor binding domain. You see in these different colors, all the mutations selected, or let's say indicated based on the fact that they are either shared by all the Omicron variants or only by some of them. And these two are specific for the variant Omicron 4 and Omicron 5. And they are located within this green area, which is the surface that is physically interacting with the AC2 receptor. Here above, again, just to give you an idea how many amino acids have been changed in these Omicron variants. It's really amazing. But the point is, what is the role of these additional mutations that make these variants even more contagious than the previous Omicron lineages? And this we were able to learn by actually studying the mutated viruses in terms of response to antibodies and ability to engage the receptor. So these graphs that seem complicated are indeed very simple to be understood, because they just represent the ability of sera from vaccinated people. Those are people vaccinated with the AstraZeneca vaccine, here with the mRNA-based vaccines, ability of the serum to neutralize the virus. And these are the different lineages. This is the Wuhan strain, and then we have the Omicron BA1, sublineage 1.1, and then 2, 3, 4, and 5. Now, just to keep things simple, the lower the number here, the lower the ability of the serum to neutralize the virus. And I think it's immediately understandable that all the Omicron variants progressively became more resistant to neutralization by antibodies that are induced by vaccination. Even, in this case, more than 10 times with respect to the original Wuhan strain. And the same is true if we then test directly the virus in the presence of neutralizing antibodies, purified. And again, you can see that the numbers are decreasing. So the Omicron, all the family of the Omicron variants is more resistant to neutralizing antibodies. And unfortunately, the 4 and 5 sublineages are the most resistant so far detected to neutralization by antibodies. The reason is, as I explained to you before, the fact that many epitopes are mutated in this virus that are recognized normally by antibodies.
Giovanni,
yes?
Sorry if I interrupt you, but if you would come to maybe...
Yes, that's actually the very last
Yes,
last
thank
slide.
you very much, so that we can switch to the other presenters. Okay. Thank you very much.
So the virus is more contagious because it has a higher affinity for the AC2. These are, just sensor grams for a particular machine, but these are the numbers that are interest you. The lowest, the number, the highest, the affinity. And you can see the 4 and 5 Omicron variants, they have the highest affinity for the AC2 receptor. And this because those additional 2 mutations are, changing the electrostatic surface that is interacting with the AC2, making it more complementary to the receptor. This is a new variant that has been just detected a few days ago in India, and it might even be that it can become more contagious than BA4 and 5 because it has this purple, a large number of unique mutations whose significance we are still trying to understand, but is again a proof of the high genetic variability of the virus. Okay, so just let me conclude with what could be an explanation of this evolution. Well, just to keep things very simple, the more the virus is replicating, the larger is the size of the progeny, the larger is the number of mutations. When it is confronted by the immune response, it depends on the strength of the immune response, the ability of the virus to mutate, which is represented by this evolutionary potential here. So those people that are not completely immune, meaning exposed to previous variants or maybe with the non-immunocompetent system, they are delayed in, mounting a very efficient response against the virus, which is still able to replicate, but also has more time to mutate. So this is the reason why, as soon as a variant appears that is resistant to our immune system, it drives the evolution of new variants. That's a phenomenon that will continue unless we will find a way to build a very strong immune response in all the people potentially exposed to the virus, giving the virus less time and less potential to evolve. So sorry if I was, too long in my presentation, and thank you very much.
Thank you very much, Giovanni, because of this very competent but also very clear overview and explanation. I believe that everyone has appreciated. So now I would like to leave the floor to Armando Gennazzani. Armando was here with us this morning, but because of the many commitments that he has, we heard that he also be part of the many regulatory committees, so he had to leave, but he wanted to be with us. And so I'm forwarding to Armando some questions which are crucial for me, but I believe also to many of you. So just following what we have discussed so far about the protection that we have with the, against the new variants by the vaccines. So my question now is to you, Armando, in 10, 15 minutes. I know it's a difficult task, but does the world still need a new COVID-19 vaccine? Or maybe, do oral COVID antiviral drugs offer new promise? So these are mine and the question also from the audience. Thank you, Armando.
Thank you very much, Maria Laura. Can you hear me?
Yes, we can hear you.
Okay, perfect. So I think I will speak less than 15 minutes, and I don't have any slides. And so I will also steal 3 of my minutes to say something different. So I think evolutionary virology is a fantastic field. So I really enjoyed Giovanni's talk, which puts things in context. And at the same time, it allows us to see evolution going at a very fast pace. And with this introduction, I would like to congratulate you for having become president of the division. I forgot this morning. And I would like to also to congratulate ESMEC for being inclusive. I've met Stephanie, part of the board of the school. I've met Tracy. I think inclusiveness in this particular era cannot be given for granted any longer. And so I am really happy about this, and I'm really happy to use 2 minutes of my time. Okay, so instead, regarding regulatory and vaccines, the question is, do we need new vaccines? This is a very important question, and not so obvious. The first thing is that we are overall all vaccinated. All those, let's say, in wealthy countries which wanted to be vaccinated are vaccinated twice, 3 times, 4 times, in some countries 5 times. So we definitely do not need a vaccine for the primary immunization. For companies which are developing those, then do not find what we said this morning, and hello to everybody, it's still me, the one which was present this morning. They cannot find an unmet medical need. We do need, though, boosters, this is for sure. And the question, and following from Giovanni, is what sort of boosters do we need? Do we need boosters which just increase the immune response we've generated with the primary vaccination regime, or do we need variants? Well, I think we need both, and I think in September, possibly the first week of September, the EMA will authorize the first variant vaccines. As Giovanni has said, the vaccines have a very, let's say, strong mutational burden. They are starting to go away from what they were originally. So we will have new vaccines. A question, and welcome to uncertain times, is what this virus is doing. Is it increasing its contagiousness by it becoming, let's say, less dangerous? Or are we not seeing it dangerous just because we are all vaccinated? And this is something that really, it's a matter of having a crystal ball to understand if this vaccine is going to become like a flu, sorry, not this vaccine, this virus is going to become like a flu, which will stay with us forever, or we will see the worst has still to come. I think we all hope the first one, and the first signs that it is this way are on the way and are here. But it's wishful thinking. What other drugs do we need? Well, we definitely still need drugs which can be used early on when someone gets ill. This also reduces the amount of propagation of the virus, the infected people, and reduces the people which move on to, let's say, more severe COVID, although these are fewer. We have a few antivirals. These are Paxlovid. It seems to be working. We have Molnupiravir, which we don't really understand what it's doing and whether it's working or not, and why the data was so clouded and confused along the way. We definitely need more antivirals which will cover variants that they come. And I think companies are working on trying to achieve those. Again, Molnupiravir, independently of how it works, and Paxlovid, were fantastic feats in 2 years that they managed to do. So I think those we still need. Do we need instead antibodies, monoclonal antibodies, which we can inject in people which are which have a low titer or are seronegative? Well, we would need them, but it seems that we can't develop them fast enough following genetic evolution of the virus. So as soon as we get monoclonal antibodies on the market, they start becoming, let's say, old, because they are unable really to protect from the viruses. We would need some antibodies which we can use in prophylaxis, for example, as well. And we possibly still need drugs which can affect the decourse in hospitals when the patients develop a more severe form. So I think this is the scenario. I hope I've not been too fast, Maria Laura, but I think most of us have heard about vaccines and drugs for the last 3 years. We really can't bear hearing other 20-minute talk about them. I think companies are doing a great job, and more drugs will arrive. We have drugs now that we can use in outpatients, we can use in inpatients, we can use in ERs. So the armamentarium is increasing. It's looking good. With the new vaccines, it will get even better with the variants. I believe, although it's just a prediction, it's personal thought, we will all get vaccinated again before the fall. I think this is something maybe that people wanted to know. So this is my impression. We will boost the antibodies, hopefully against the new variants, as Giovanni said, and hopefully we will have a good autumn and we will sort of put behind the experience we've had with COVID. Thank you, Maria Laura.
Thank you very much, Armando. I'm also very, very glad that you find the time to participate to this roundtable. We truly appreciate, and also I have also enjoyed your even a kind of optimistic point of view, and I would like to highlight what you have already said, but you are a pharmacologist, we are medicinal chemists, but we should have in mind as first that we should put efforts together, especially because we are in academia, and so to consider as first the unmet clinical need and look for that and put all the energy for that, and especially do it together, okay? Because both, as we medicinal chemists, you pharmacologists in Italy, it's a kind of different profession, but we both love drugs, and so I believe that working together now it's even more important than before. So thank you very much also for that, for participating and for sharing with us your expertise and also your privileged point of view. So thank you very much. I know that you have to go. We see that you are in the car. So maybe we can, again, thank you because we know that you are leaving. Grazie, Armando.
Enjoy tonight.
Thank you. Okay, so let's move now to the next speaker. Again, maybe always repeating the same story, but I'm truly happy to have Carmen Hill with us today. Do we, okay, no? Oh. No, please, you can upload the presentation. Carmen is a friend of mine.
I want to share the screen.
You want to share the screen here? But full screen mode, okay, thank you. So Carmen is here on top. Carmen comes from Centro de Investigaciones Biológicas in Madrid, in CSIC. We know each other since many years, since I was in Madrid in 2009. And so it's a pleasure to have her here, but then you're wondering why Carmen is here, because she's a friend of Maria Laura? No, there is a reason. Because she was very active in the COVID field, and I couldn't congratulate, so I'm saying this now to everyone, but she was so good because just after the outbreak, she was able to publish a perspective in Journal Medicinal Chemistry. Everybody knows Journal Medicinal Chemistry, including myself, of course. And she was so good and published a very nice perspective. And I'm suggesting you to read it because together with Anna Martinez, they gave a very nice overview on the possible targets for medicinal chemists to address. Okay, so I enjoyed that publication a lot, not only myself, because so far it has already, I checked the 250 citations, so a very nice one. So I'm very eager, Carmen, to see your point of view as a medicinal chemist about what we can do in the discovery of small molecules and the targets and the opportunities.
Okay, thank you, Maria Laura, for your kind introduction. It's true that we are friends, but we are also colleagues. And so, okay, it's really a pleasure for me to be here to present, or better, to give you an overview of the different drug targets and potential small molecules to treat COVID-19. As Maria Laura said, since the very beginning of the pandemic, and myself decided to see what we can do to fight against the pandemic, because I think that in this unprecedented situation, it was clear that, at least from my point of view, one of the positive things was that at that time, all the research community were working together to find a solution from our different situations. I mean, and I hope that this works. Okay, this is just to, I don't want to introduce now the COVID and the SARS-CoV-2, but this is just to show you that here, that today, the numbers are really amazing, and then the fight is not finished, and we need to continue to fight against this virus. Well, what we have done at the beginning, and I think that is just is that all medicinal chemists need to do when we want to start a new drug discovery program, is really to know who is our enemy. I mean, in this case, SARS-CoV-2 is the virus that we would like to fight against, and that's I'm sure that most of you know that SARS-CoV-2 is a coronavirus, that is our RNA virus. And the genome of this virus codifies 4 different structural proteins. And then the M protein holds the genome, and also the S, E, and M proteins together create the viral envelope. But the most famous protein of this virus is, of course, the spike protein that we have been hearing about this protein, and it's because this protein is the responsible of viral infection and allow the virus to attach to the host cells and start the infectious process. Well, in order to start a drug discovery program, what the medicinal chemistry needs is really a target. So at that moment, when we started to see what's happened with this virus, it was very important to know the viral life cycle, because in this way we can identify the different drug targets to develop new drugs. Well, in fact, if we focus first on the entry process of this virus, we see that really there is this virus attached to the receptor, the ACE2 receptor, and focus on this viral entry process, we can identify different host-based targets, because we will see how the target can be classified into types. On one hand, the host-based target, and afterwards the viral-based target. Well, the first step in the entry process is the bind of the spike protein to the receptor. That oh, sorry, because there is so much movement. Well, the spike protein is a homotrimeric protein that has 3, and the monomer has two main subdomains. The S1 that is responsible to attach to the ACE2 receptor, but before to attach to the bind of the spike to the receptor, it was necessary to be cleaved by one host serine proteins, that is the transmembrane protease serine2. And after this, the virus can enter into the cell. At the beginning of the pandemic, thanks to the work that has been done before in other coronavirus, such as SARS and MERS, there were some similarities between these viruses, and in fact also the SARS virus used the ACE2 receptor to enter into the cell, and because of this, really it was early identified. But afterwards, there are another receptor and co-receptor that were identified because they have also a role in the viral process entry. So if we see here, we have identified 3 different host-based targets. I mean, this host protease and also this 2 receptor, and focus on these 3 targets. It could be possible to develop emerging host-based therapies. In fact, we can see here in the next slide different small molecules that were identified in the early times of the pandemic, acting as ACE2-based therapies, such as different blockers of the receptor, and also antibodies, or even protein-protein interaction inhibitors, because the receptor binding domain of the spike binds to this receptor, and if it's possible also to develop protein-protein interaction inhibitors. Also, there are another therapies that were investigated looking for an inhibitor of this other receptor, the AXL, and also inhibitors of this host protease identified at the beginning of the pandemic. But also, after these initial findings, it was seen that other host proteins were important for the entry of this virus, not only the TMPRS2 protease, but also another host proteins like furin and catepsin were important for the viral infection. And also, another receptor were also important for the entry of the virus, like the neurophilin 1 receptor, and also the basicin or the CD147 receptor. And then, beside to the initial emerging therapies focused in our first 3 targets, there are nowadays also another emerging therapies based on this new target that were identified after the first times. So in this sense, there are now some furin inhibitors that showed also antiviral activity against this virus, also catepsin inhibitors or different molecules based on the antagonists of this other receptor identified afterwards. So after the viral entry, the virus of this, after this binds to the receptor, there is a receptor-mediated endocytosis, and then the virus is included in an endosome. And in this sense, here there are some different host enzymes that are important in this viral infectious process. In fact, there are some kinases like AAK1 and GAK that are related to the clathrin-mediated endocytosis, and there are important viral host targets to fight against this coronavirus. Also, there are another kinase, that is PIK5, that is related to the endosomal maturation, and also another protein that are related to these endosomal pathways, like the endosomal 2-port channel, TPC2, that have shown also to have a key role in the viral infectious process. So these different targets are also the focus of different host emergent therapies to fight against coronavirus. But also, after these initial findings, another target are really being discovered. Okay, not every day, but really this is really an evolving field. And also, it was seen the importance of the Niemann Pick C1 receptor in the viral infection process. And in total, we can see here how, if we consider all these targets, there are also different therapies that are under study, focused on these different targets. And in fact, there is one FDA-approved drug that was baricitinib, that was initially approved in November 2020. And this is a AAK1 inhibitor, but also this molecule is able to inhibit also the GAC enzyme, but also this is a non-selective kinase inhibitor, also inhibit the JNK. That means that really the compound not only affects the infectious process acting in this host-based target, but also has an anti-inflammatory effect that can also be important and good for the treatment of this patient. And just to mention that really this drug was approved alone, because before was approved in combination with remdesivir, but now the treatment alone is allowed since May 2022. And also, there are another emerging therapies based on the other different targets, and I'm sorry, but I have no time to go deeply into each molecule, but just to show you how many different small molecules based on different targets in different drug discovery programs that were really discovered during this time and that were also tested as antivirals in SARS-CoV-2. Well, and the next step in the life cycle of the virus is really the point in which the virus enters into the cell and then release the genome. And the genome, using the ribosome of the cell, produce two different two proteins that are the polyproteins that were subsequently cleaved by two viral proteins that are the PL-Pro, that is the main protease, and also the papain-like protein. And in this way, the different non-structural proteins were synthesized that finally produce the new virus that are liberated from the cell by exocytosis. Here, just to mention at the beginning, only some of these non-structural proteins were known, but nowadays 16 are known, and also you see that really there are many of them that are also known by almost everybody, and that also has led to different drug discovery programs. The three main viral proteins that has also very important. They are also very important because has led to different FDA-approved drugs. The first one is the main protease. That really has led to an intensive work from different groups from the academia and the industry. And in fact, as you know, the approval of nirmatrelvir at the end of last year was really a key point or a key achievement in the field because it was really the first molecule developed just to target the SARS-CoV-2. Because as you will see later, really another FDA-approved drugs were initially developed for other indications and then repurposed to COVID-19. Such is the case of remdesivir that was initially developed as Ebola virus anti-Ebola virus drug because this drug as a polymerase inhibitor, and now it's repurposed for the treatment of COVID-19, and also molnupiravir that was initially developed for the treatment of influenza, and that is now also approved for the treatment of COVID-19. There are here another viral protein that has led to the discovery of different molecules with the potential to be developed as anti-SARS-CoV-2 drugs. And here I would like to emphasize one point that is very important for the drug discovery field, that is not only the knowledge around the viral life cycle, but also the work done by the structural biologists group that has led to a huge number of crystal structure of all these targets that really allow and simplify or just directed the drug discovery programs. And here, just as a conclusion of this quick overview of the different targets and drugs available for the treatment of COVID-19, this is really the picture of the life cycle when we published this review that Maria Laura has commented, that was in the first half of 2020. But really what is important, and I would like to emphasize it, as really today we need an update of this life cycle, and there are much more knowledge around the different targets, and there are a new emerging viral and also host target that can be used to develop new drugs. And here, just to show also the drugs that have been approved now and the different point of the viral cycle in which they are working on. And finally, I think that's really this slide probably connect with the next intervention, just to show you and to remark that really from the 4 drugs that we have today for the approved for the treatment of COVID-19, 3 of them are drug repurposed drugs because also this morning it was commented during the rare disease session, is really the faster way to have drugs in a short time for a really unmet disease, such as was the pandemic situation. But also what is important is really the release of nirmatrelvir, because it was originally developed or just for the treatment of COVID-19. And finally, I would like to thank really to all the research community from the academia and also from the industry, because working together is the way in which we can really have a solution to this problem, because it's not only the work from the medicinal chemists, but also from the pharmacologists, from the molecular virologists, and also the structural biology. And that's all.
Thank you very much, Carmen, for this very inspiring overview. We have a lot of food for thoughts as medicinal chemists, a lot of target, but we know that we can look at your perspective, find more details. But in any ways, yes, there is a lot to do, even from our side. And now I would like to go to the next speaker. Next speaker is Franco Gillucci. He's not a medicinal chemist,
no.
So
much better. I'm an intruder.
Much better, as we said before. We need this kind of contamination among disciplines if we want to bring a drug on the market and if we want to do something great. And because he is, I want to be precise, he's a hospital medical advisor at Pfizer. So he's here because he can talk a little bit from his point of view as a clinician about the very nice story of Pfizer pill, because we can work not as a medicinal chemist, but common person, no? But Pfizer COVID-19 pill, and even the clinical effect. So,
thank you.
Please.
Thank you very much. Good afternoon. For me, it's a real pleasure to be here and have the possibility to share with you some information in this really interesting scientific section on COVID-19. Probably it is more easy after the the previous presentation that it is very, 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 very wonderful. Thank you for the introduction. I'm Franco Gillucci. I'm a medical doctor. I work in Pfizer as a medical advisor with particular focus on COVID-19 and new therapy for COVID-19. Considering my background and my nature of clinician, I want to start sharing with you some information about the clinic of COVID-19. As we know, COVID-19 is a disease related to the infection of SARS-CoV-2, a coronavirus emerged in China in the end of 2019 and beginning of 2020. And all we know that during the 2020, this infection evolves in a pandemic for the whole world. And we know how, we know the weight that the pandemic has still in our life, in our daily lives. COVID-19 is a disease with an heterogeneous manifestations. It can involve different organs, so a multi-organ involvement can be possible. And we have some classical symptoms, such as cough, fever, dyspnea, myalgia, but we have also some atypical symptoms, like, for example, gastrointestinal disorders, articular pain, fatigue. And this type of symptoms can complicate the diagnosis. And after 2 years of the pandemic, all we know the importance of the early diagnosis, because with an early diagnosis, we can early approach the disease with all the instruments that we have today to challenge this pandemic. Classically, according with the difference that can be present related to the different variants, the disease can evolve in different stages, from an asymptomatic stage to more severe stage of the disease, in which patient can be critical. And in this evolution, an important role is played by our immune system, because in the early stage of the disease, the immune system has the role to challenge the disease and the diffusion of the infection in our body. But with the amplification or the activation of the immune system, it is possible that the same immune system can cause damage at different level and in different organs as a consequence of an hyperactivation of the immune system through cytokines, interleukins, and so we have an unbalanced system in this sense. And this is what happened in many critical patients, in which the evolution mainly is the presence of pneumonia and respiratory failure, with all the consequences that all we know. Considering the unpredictable nature of SARS-CoV-2, today we should think about two different strategies to challenge this infection and this virus: a prevention and a treatment. And in this sense, we have today vaccines for the preventions and specific treatment to treat the disease in the early phase of the infection. So, specific treatment for the acute infection. In this sense, since the start of the pandemic, Pfizer has committed to delivering both treatments and preventive solution to address the COVID-19. And today we have instruments in this sense, vaccines and specific therapies. About specific therapies, as company we have a specific antiviral treatment. This antiviral treatment is called Paxlovid, and it is an association of two different molecules, nirmatrelvir, that is the molecules with the direct effect on SARS-CoV-2, so the molecule with the antiviral effect, in association with ritonavir, that is used as a PK enhancer to guarantee the good plasmatic levels of the active molecules to inhibit the replication of the virus. The target, the specific target, as mentioned in the previous presentation, is the main protease of SARS-CoV-2, MPRO, called also 3CL-PRO, and the effect is the inhibition of the activity of this enzyme and then of the process of proteolysis that is essential in the infected cells for the virus to realize structure with the aim to replicate itself. So the consequence of the block of this specific enzyme is the block of the replication, and so the incapacity of the virus to create new variants and so to give diffusion of the infection. We know, in fact, that our aims in the management of this type of disease are different, and these aims are based on the control of the infection in terms of diffusion of the virus, but also to prevent severe form of the disease, prevent hospitalization, prevent death, and also the consequence in long term of the disease, because we know that another important point related to COVID-19 is the long COVID and the consequence in the long term in patients affected by this type of disease. Paxlovid is approved by FDA, was approved by FDA to treat adult patients with confirmed COVID-19 infection, mild to moderate COVID-19. and pediatric patient, 12 years age or older, with at least 40 kilograms of weight. And after the approved by FDA, the CHMP of European Medicines Agency approved the use of Paxlovid, of our molecules, to treat adult patients with a confirmed diagnosis of COVID-19 who do not require supplemental oxygen for COVID-19 and who are at risk of evolution to a severe disease. This is, in fact, the target of this drug. Patient at risk to evolution to a severe form. What it means? Risk factors associated with the evolution to a severe form of the disease are essentially the presence of comorbidities such as cardiovascular disease, pulmonary disease, renal disease, diabetes, old age, and the presence of this risk factor can represent a problem in terms of evolution of the infection in a patient. So a patient with this type of characteristics can evolve in a more severe form of the disease, and our molecules demonstrate a great effectiveness in this type of patient. The results in terms of effectiveness derives from a specific trial, APIC-HR trial, and the cycle-HR stands for high-risk patient because in this trial we enrolled patients with risk factors for an evolution to a severe disease. And very, very important results show us that with a treatment with our antiviral, patients have less risk to evolve in a severe form. In the specific, considering the two arms of the trial, so patient treated with the antiviral and patient treated with placebo, the results showed a reduction in terms of the primary endpoint of the trial, that is the reduction of the risk of COVID-19 related hospitalization or death for any cause in a period of 28 days, a reduction in terms of percentage of 89% in patients treated within 3 days from symptom onset and 88% in patients treated within 5 days from symptom onset. This is another important point. As in many viral disease, the early treatment is fundamental to have a big chance to have effectiveness treatment. So in this sense, also the early diagnosis obviously is fundamental. These data are confirmed recently by the final clinical study report that confirm a reduction in terms of risk for this outcome of 86% in patients at risk treated. So very, very robust results in terms of effectiveness. Other important results, for example, are also the results in terms of safety, obviously, but also, for example, in terms of viral load. Patients treated with antiviral showed a reduction in terms of antiviral titer of tenfold considered in patients treated with placebo. So at 5 days from the treatment, there was observed a reduction in the titer of the viral load related to the nasopharyngeal swab. And this is the expression of the efficacy of the drug in terms of block of the replication. Regarding safety, the APIC-HR trial showed us a substantial, similar trend of safety in terms of patient treated with the molecules and patient treated with placebo, and in particular related to adverse events. 23% of presence of adverse event in patients treated with the molecules and 24% of adverse event in patients treated with placebo, so a similar trend. And regarding severe adverse events, 1.6% in patients treated with the molecules, the active molecules, and 6.6% in patients treated with placebo. So a very strong results in terms of safety. And the same trend was observed also in the adverse events related to the discontinuation of the drug. In this case, 2.1% in patients treated with the antiviral and 4.2% in patients treated with placebo. So these results show us a good profile, not only in sense of effectiveness, but also in sense of safety. So today we have this type of therapies to challenge the disease and to change the evolution of the COVID-19 pandemic. And as company, Pfizer is in collaboration with country governments and global health organizations to break barriers for the access to vaccines and to therapies. The aim is to realize an equal opportunity for all the people to access to this new treatment and to these different strategies to prevent the evolution the pandemic. To June, for example, I can share with you then that more than 12 million of treatment were supplied, and more than 1.5 patients at risk was treated with our molecules. And it is a big result in terms of what the science can do with world collaboration to address an important health problem such as the COVID-19 related pandemic. And with this, I conclude. I'm very proud to be able to share with you some information related to our molecules, but in general about the strategies to address COVID-19. And I want to thank you very much for the attention and for the opportunity. Thank you.
Thank you very much, Dr. Gillucci. I was very glad to hear about the safety and effectiveness of this drug, and especially backed up by data, which is the most important thing. So thank you also for providing your point of view as a medical doctor, and also for touching the very important point about the access to the new drugs, which is to me and for all the society a main concern, because we should not only think about the situation in Italy, okay, here we are in Italy, and many of the actors are from Italy or from Europe, but we know that the battle will be won if we will eradicate the virus from all over the world. So thank you very much also for briefly addressing this point. We don't have time to discuss, but because we are still a little bit late, and so I would like now to go over Carmine Talarico. Carmine Talarico is a medicinal chemist, so and he has participated to a very important project. I have to confess that I was very proud when I read the news release about the fact that Dompe was leading this important European project termed Escalate, if I'm not wrong, and because, of course, a bit proud because as an Italian, and also I believe that your experience will be very well appreciated, because you can show us even how the use of big data, artificial intelligence, whatever, could be very useful in addressing drug discovery. and maybe we can understand even a little bit more about how it is practically possible to use this new approach to make new drugs against unmet medical needs. Okay, thank you very much for accepting our invitation and joining the ecosystem, you know, medicinal chemistry in Italy.
Thank you so much, Maria Laura. So first of all, let me thank, I don't know why, maybe it's, oh, yeah. First of all, let me thank Maria Laura and the organizing committee to this kind invitation. It's an honor to me to be here with all of you today. So today I'm gonna talking about high performance computing and artificial intelligence and big data in search of new drugs to fight COVID-19, and in particular. Maybe it's too far. Okay. Particular, I'm talking about the Escalate for COVID project, and as you already know, in the end of 19, the first class of cases of pneumonia raised up all over the world, and in March 2020, the WHO declared the status of pandemic. So, in particular, in I don't know. Okay, thank you. Yeah. In particular, in February 2020, Dompe organized and participate to this European Horizon 2020 call dedicated to the response to the COVID-19 with other 17 partners coming from 7 countries all over the Europe. And the project in particular starts from started in April 2020 and ended in September 2021. The main objectives of the project were to identify molecules capable to targeting the COVID-19 and coronavirus in that specific case, and to develop effective tool for countering future pandemics, and also to exploiting the European supercomputer capabilities and infrastructures. Next, please. So the partners that composed the consortium had different kind of expertise in terms of drug discovery, structural biology, but also HPC and managing big data. And to support the consortium, we also had, next please, what we call league, that were external collaboration that supported the project during these 18 months. Next, please. In terms of European High Performance Computing Center, the project was supported by HPC5 from ENI, Marconi 100 at Sinega, but also from other HPC centers around Europe, such as Mare Nostrum in Barcelona and Eureka in Germany. Next, please. So in this slide, you can see some numbers with respect to the results we achieved, and in particular, the huge amounts of experimental data we proposed and validated, such as experimental biological and phenotypic assay, but also huge amounts of in silico data, always supported by scientific production and paper production. Next, please. So mainly the Escalate for COVID workflow was divided in two main phases. Phase 1 was the immediate response in 15 months, and what we call the phase 2, on bottom of the slide, there is the de novo part that is still ongoing beyond the duration of the project. Next slide, please. In particular, for the immediate response, we tested more than 10,000 drugs coming from different library, selected and ranked by using in silico methods, and these drugs were tested over more than 10 different assays, phenotypic, biochemical assays, and at the end we produced 3 preclinical POG, arrived to the best one. What we propose is the raloxifene as a possible treatment for polysymptomatic COVID-19 patients. Next, please. So, as I already said, all the results we achieved were always supported by high-quality scientific production. We publish our results on very important journals belonging to the Nature group, Lancet group, but also we were glad to add also some journal cover, and all the publication goes from the, for instance, in silico validation till the results obtained during the clinical trial. Next, please. And again, to support the scientific community, during the project we also developed some web portal that was useful to give access to the scientific and to the researchers to have access to the data we produced during the project. And we open, let me say, these 6 windows on the data produced, and there are, of course, still updated now beyond the project. And if you're interested in some of them, you can visit the escalate4covid.eu web server. Next, please. And in particular, among these websites, we are so proud of the spike mutant website, in which we release 3D structural information, such as 3D structures, but also molecular dynamic trajectories, in order to understand the structural environment of the spike protein. In that case, here I reported some 3 publications in which this research group used the data we released in the website to carry out their own research. Next, please. And again, in terms of open science, during the Escalate for COVID project, we also launched the Mediate initiative, that is a crowdsourced virtual screening collection from computational and chemistry groups and AI and machine learning communities, and in which the researchers can download 3D structure of the main viral proteins and 3D dataset in order to make their own molecular docking simulation. Next, please. This is the scientific advisory board of the Mediate initiative, and we were so glad when Ade Warshel, Nobel Prize for the Computational Chemistry, joined our scientific advisory board, together with Professor Apostolov, Ted Ko, and Ye Yang from Shanghai Institute of Materia Medica. Next, please. This is one of the last slides. In the slide, it's a comment on how the HPC infrastructure is supporting the pandemic. In particular, in that case, in November 2020, Escalate for COVID performed the most complex HPC virtual screening experiment to identify new therapies by running in parallel on 2 HPC centers in Italy, the HPC5 from Eni and Marconi 100 in Cinega. In particular, we virtually tested 70 billion molecules on 15 active sites of the SARS-CoV-2 proteins by reaching 1 trillion of molecules docked in 60 hours. And this was possible also thanks to Politecnico di Milano that allowed to our proprietary virtual screening software to run on HPC infrastructures, but also to SaaS company that allow to manage the huge amount of data we produced. All the information and the data we produced during these experiments were then released on the Mediatek web portal. Next, please. This is the last slide. So the lesson learned here is that advanced technologies for real is speeding up the discovery of new treatments. Advanced technologies, not only in terms of HPC infrastructure or huge calculation, but also technologies applied to the experimental validation. And in my opinion, create networks of collaboration is mandatory in that sense, because give us the opportunity to access very quickly to expertise that not all the groups had in-house, of course. We confirm how the drug repurposing approach is still valid in the infectiology field, and with respect, in particular for the Escalate for COVID project, we confirm how raloxifene showed an optimal activity profile to advance in COVID-19 indication. Okay, thanks a lot.
Thank you very much, Carmine, for showing us how is it possible to exploit HPC, AI, data, and especially for showing us a fruitful example on open science. I'm a particular fan of open science. Someone is skeptical, you know, is it possible to work in an open science scheme with companies? Yes, it is. If everyone is very committed and convinced. So thank you very much also for giving us a lesson. We learned a lot about with this project. So now it's the turn of Christian Montalbetti. Christian is the director of medicinal chemistry at ERBM, or if you want, IRBM, but in Italy it's ERBM. And so again, I'm very glad to have Christian here. This is a contact, very new, but we have already exploited some synergies so far. So, Christian, I'm very eager to see your experience. I know that you cannot share too much at the moment, but if you can give us the flavor of what you are doing at ERBM, we'll be very glad. Okay, thank you.
We're going to see that. So first, thank you very much, Maria Laura, to have invited me, and also to all the team, because the hospitality has been world-class. And so I'm also very happy that I didn't use your paper, carbon to do the, because I also wanted to do a review on what kind of target we can address as a medicinal chemist, and I would have copied your paper, and so I've done it totally different. I read it a time ago, and I didn't reopen it. So maybe we go to the old school next slide. Okay, so it's a very intense field of research. So if you think it's only 2 years, basically, that the research has really started, because the time that you start a project, that you form the team, 2 months were gone. So it's basically 2 years. 2 years, there's multiple vaccines on the market. There's now small molecules on the market, and we had a fantastic lecture on actually how it works in the clinic. And it's really a big achievement for the European community, and I think that maybe it's going to be in the next session, but it's an extremely fantastic achievement worldwide. So next slide, please. So if we look at, I used the Cortellis database to see a bit what are the activities in the field. So you can see that a lot of activity is related to vaccine and antibodies, and we heard about them and what they can do, messenger RNA, adenoviruses, inactivated viruses now. There's also in the peptides with recombinant peptides that are used as vaccine, cyclic and linear peptides to interact with the diverse surface molecules, proteins that are there, receptors. So I'm not going to look into that. Then there's, they have a category of other. So you have Herbal, Polymers, drug conjugates, nitric oxide, for example, you spray nitric oxide in the nose when you have the infection, kills the virus. But so that's not really the, I think, the juicy, interesting part for medicinal chemists. So next slide, please. So here it's really what we are interested in, and it's the small molecules. So, and also they have a way to divide the small molecules in small molecules and a bit bigger molecules. But never mind. So I'm going to start with the blue, which are the a bit bigger molecules. And here we can see ivermectin and azithromycin. Those are big molecules that have been discovered by more serendipity, population that had COVID coming, but they were not affected seriously. Why? Because they had received those drugs. Having said that, the World Health Organization says that we shouldn't use those drugs. And they even warn that please don't use the veterinary form of those. People are really desperate sometimes and use whatever they get their hands on. So you have natural products and Protacs, for example, that are developed. And then if you look at the small molecules, for example, the Pfizer one, Nirmatrelvir, was very interesting because it comes as a single molecule and still stuck in phase 2-3. But then you have the whole story. It's if you put it with another protease inhibitor that doesn't play the role of a protein inhibitor, which is really, I think, a bit of a nice story. I don't know, I like that. And it becoming allows the drug to get the concentration that you need on the cells and then to work. And so it's really something that is a fantastic achievement. And also plays that ritonavir can be used because when you go to the MPK and tell them, oh, my drug is not going in the cell, can I use it with ritonavir? Oh, no. Here we have a proof it can be used. Okay, in man, safely. Good. So looking at the, strange, the title has disappeared, never mind. So here are the drugs that are basically in clinical. So 4 have been launched, okay? 2 are undergoing registration. I'm going to go in details then. And so in the launched, you have the Fluripavir, but it's in India and Russia, so it's been very limited, so I don't think it's the best drug ever. The Remdesivir, that was launched early on by Gilead, and that is not pursued as a very, very effective drug. But I was surprised that it received this year authorization for pediatric use in COVID-2 situation. So it's still kicking on and going. Then there's the molnupiravir, that is, all those are nucleoside, and they are hitting the polymerase. And you can see that there are other drugs that are still coming on. For example, that the Chinese drug at the bottom, that is interesting because it's basically another prodrug that will, of remdesivir, and it has adulterated deuterium now in a position, so maybe they wanted to block the metabolism in a specific position. But that is going in clinic, is in registration, so the final step. But in Uzbekistan, again, then Chinese sometimes choose the place where they want to do the studies. So we have already seen Paxlovid, and what I wanted to show you is another drug from Japan, Shinogi, that makes that drug, that is also now an SP5 inhibitor. And what is interesting, it's a triazine-based, or if you want to get scared, cyanuric acid-derived compound. As it's substituted on the nitrogen, I think it should be totally safe, of course. But so that's interesting because we are getting away now from the amino acid derived or modified amino acid peptide type of world. Okay, next slide, please. Oh, okay, so you have to do 2 clicks. Okay, so here I wanted to do a bit of what are the hottest targets at this point. Oh, I forgot to mention on the previous slide, so that if you can quickly come on back on the previous slide. Yes, here. So there's 37 small molecules that are going clinical trials as we speak, so that's from the database, so maybe 1 or 2 more. Maybe I missed some that are in combination, because the combination are dealt separately. 26 of which have already been launched. So, and also quite a lot of them have higher phases in other indications. So that means that it's really repositioning. But honestly, in 2 years, if you think the time that it takes to go when you have a preclinical candidate, and you want to make it the preclinical compound, you have to go through all the non-GLP tox, the scale-up. So there's nearly 6 months for a very, very big company as Pfizer. Maybe they can push it. For a medium-sized company, it's 1 year solid work. So even if you imagine the time that it took, even if there was a project that was already there on MERS or on SARS-CoV, to be able to repush everything, design the clinical phase, it's a fantastic achievement. Okay, next slide. So let's talk about the targets. So the 2 king or queen targets, I don't know how to call them, but there's the NSP12, of course, the polymerase, and NSP5. So for the time being, the polymerase has been addressed with nucleoside, and non-nucleoside are still in the pipe, or hopefully they will come. And we see that so far, it's not the best strategy. NSP5, we've seen that there are drugs on the market now and coming, and NSP5 is very interesting because you can target it via covalent drugs. Okay, the catalytic stuff is a cysteine, 145 if I'm not wrong, and the main protease also cleaves peptides at a very specific place. So I forgot now the amino acid, but the human proteases, and there are quite a few circulating or in the cells, are not cutting peptides at this place. So that's a very good opportunity to have selectivity and hence safety. And then also I read that NSP5 is involved in the immune response, not the immune, sorry, inflammation response and the cytokine storm that happens afterwards. So it also, by inhibiting it, you also avoid the inflammatory response, which is responsible for the death of people at the end. So So the little sister, or should I say the big sister, ANSP3, because it's a very big protein, is the papain-like protease. Has also a cysteine there, but here it's a triad, then in the polymerase is a dyad. And so people started to work on that one, and also doing dual inhibitors. So there's work going on, but it's not as worked as the NSP5, of course. There are some PPI inhibitors, for example, spike-ACE interaction with small molecules. NSP13, we've seen lumafactor, but there's also very little description there. But so we are interested in NSP13. So, in terms of the human targets, we've seen that so it's a non-exhaustive list here, but what you can see roughly, you look at them and you say, oh, it looks really like a kinase, not kinase, and you have nuclear receptor, you have apoptosis inducers, and so on, so on. So you think at the end, yeah, it looks like a bit of cancer indications. And for me, I'm a chemist, so I like to make simple reasoning. For me, it's not surprising that a virus needs to enslave the cell so that it keeps on working, like cancer. And the cell has a lot of mechanism to shut down, to stop working when something goes really seriously wrong. But cancer and viruses can overcome this. So it's not surprising that some of those targets are here. There's also then inflammation with IL-6, for example, there. So this molecule, I wanted to show you that molecule is in phase 2. So it's a cholesterol ester transferase compound that has been pushed in clinical, but it's a prodrug. So if you remove the thioester, you have the thiol. And the thiol just goes, but that's in a paper by docking, but just goes on the cysteine and makes a disulfide bond. And that drug works, it's like a covalent inhibitor, so it stays there, it's very active. So it's a way of moving away from, also from the peptide type of molecules, and there's also selenium drugs that are tested, advanced in that field for both proteases. Okay, next slide, please. So, what do you want to do? Which target? Which strategy? So, are you going for a viral target or for a human target? Viral target, you should have selectivity, better safety, but we've seen that it's also a nice strategy to attack the human targets. Do you want to target the viral cycle or or stop the lethal aspect of the disease? Of course, the viral cycle is more efficient because then you reduce the amount of virus and of also, how do you call that, diffusion of the virus. Acute versus prophylactic. Can we imagine making a drug that is prophylactic for HIV? There's treatment for people who want to have selected a certain lifestyle, and you can take that drug that is basically taken all the times to avoid getting HIV. Can we imagine having a drug that would be used, could be used all the time, or when there's one of those peaks, you distribute the drug? That could be also a way of reducing the amount of virus circulating. Validated versus novel target. So here it's a bit like going to the bank and asking for an investment. They ask you, what is your risk adversiveness? So you have to say, I like risk or not. Here's the same thing. Novel target, of course, are far much more risky because you need to validate the target, and there's a lot of years that go by before you can move into clinic. And so that's very valid if you have in-house assets. So drug that is already there, collection of compounds for the same family, and it's totally, I would have done that having a collection of or project going on in the field of or you have a target, you have, for example, I don't know, an ERK inhibitor, and you see that ERK can be used, you test it and you try to push it, of course. Then it's the NSP 5 and 12, and Carmen has a bit touched base. Can we target the other ones? For example, NSP 13 helicase has multiple functions, so can we target it via enzymatic inhibitors? So the TPAs, the unwinding, can you block that? Can you also target the drug, the protein via degradation? Can we do a PROTAC? But not a PROTAC that is also an inhibitor, because I always ask myself, why do you want to do a PROTAC if you have already a very good inhibitor? So here you would have the reason, because I worked on helicases on HCV, which is the same protein as the protease, and we failed. So we managed to get the protease inhibitor right, and that got sent to Roche. But the helicase, we had to stop the project. So helicases are not easy to target with inhibitors. Maybe we can target them with a degrader. So NSP3. NSP3, if you look at the protein, it's long like that, and in the middle you have the protease domain, and you have plenty of other domains. Is the protease domain the only thing that you can target? Can you target the others? Those are open questions. Can you find some mechanisms that are specific to the coronavirus? And there is, for example, this continuous RNA transcription. Can you target that? I don't know. And how, I don't know. So, but that's a lot of biology work that has been done to see if we can address that thing in a way, and finding if you want to attach or target the RNA, or you want to interact with proteins that interact with the RNA. Okay, next one. Okay, so we've seen the strategy, repositioning, me too. De novo hit finding, there's HTL, DEL, and FBS. So fragment and DEL allowed here to eventually find binders. So that goes towards the degradation route. Virtual screens, of course. At the beginning, a lot of paper came out, virtual screens on the homology model. When the structure came out, homology on the real structures. And then the paper came out as fragment screens and other things. So it's a very valid way of doing. And we had this fantastic talk about artificial intelligence. Inhibitor, degradator, PPIs, and open the door basically to other targets. And also explore not only the obvious domains of the proteins, but also the other domains that can play a role in PPIs or whatever. And that's the biologists, of course, that have to find out the biology. So I hope I gave you a bit of an overview of the different aspects that can be kind of like envisaged to fight COVID. And I think what is very important, you know, the obvious, and that's not the obvious, it's not the critic, but the easy, obvious stuff has been done. It's very difficult. Now we have to think, so, you know, making other, I don't know, nucleoside, maybe it's not going to help to move forward. We maybe need to do something else. And also combination will come into play. We've seen already one, but maybe different targets of COVID. And also aim eventually to targets that can be used in the future COVID. We have COVID-19, we've seen all the mutations, so a bit scary even if the message was rather positive, but when we see the rate, everybody has COVID nowadays, so it means that the virus will mutate at an even higher rate because it's in the population. So it's definitely we need to build the arsenal. Thank you very much.
Thank you very much, Christian, for this critical overview, for sharing your reflection from a drug discovery point of view. So thank you very much. We are a bit late, as usual, but there is a lot to discuss, and I didn't want to stop you because all your talks were so interesting. So now we will change a little bit the focus, sorry, because I am going to introduce you Professor Rui Moreira. Professor Rui Moreira comes from University of Lisbon, but today is here as the president of the European Federation of Medicinal Chemistry, and we are so glad to have Rui here, another friend of mine. We have done several things together at the FMC, and so we share views and perspective and directions, and today what I was asking to Rui is something different from the other speakers, and so as a president of a division, what is the role of a scientific division in these quite strange and hard times? So, Rui, please.
Thank you, Maria Laura. So it's a pleasure to be here, and my role will be different from the other members of this panel. So basically, you asked me to talk about the role of scientific societies, and in particular EFMC. So societies are indeed the platforms to convey scientific information to their members. And of course, information can be conveyed through papers in journals. But I think what societies can do is, in fact, bring people together and discuss ideas and to promote interactions, and that's the ultimate role of the societies, and that's the case also for EFMC. So I would divide, as you suggested, my presentation in two parts. So what happened in the pandemic, so in these 2 years, and what will be the roles of the societies in the post-pandemic era, if there is a post-pandemic time. So that's a big question, of course. So the first thing is that to promote interaction and discussion of ideas, I think if I could have the best example, it's exactly what is happening today. So I would like to congratulate you, Maria Laura, for selecting the people and to allow to have different backgrounds and to present the different views of how the research on COVID, on SARS-CoV-2, as you wish, has developed in these 2 years. I think that's one of the most important things. So it's an idea that we should always follow. So in this, and everything started in early 2020. Of course, there was no meetings. So meetings are the main source for exchanging ideas. So the meetings could not take place. So societies, most of the societies had to go to other options. So what EFMC did at the time, not being able to organize presidential meetings, was to go through the young scientist network. So young people from different societies across Europe, they have organized the workshops online. So the Medicaid buy online, that probably you have heard. It was a less resource that we had to use, but in fact what we have learned from it is that this type of approach has arrived and will stay on in the future. And why is it important? So it's not directly related to COVID itself in terms of conveying science about COVID and SARS-CoV-2. It happened also, so that was part of, or at least some of the topics that we have covered in these workshops, but also to allow science to continue in other topics. Of course, there are other diseases that continue to exist. There is science going on, and the roles of the societies like EFMC is exactly to allow, to give field and time to all the therapeutic areas. That I think is very important, and that is the role also for these coming years. Of course, we can, EFMC is also continuing to give the stage for the new science that is coming up related to SARS-CoV-2 and COVID. That, of course, that will continue. But one of the things that I really I appreciate about these type of discussions is to bring topics that for some of the people involved in science, eventually particularly the younger ones, is what are the characteristics that we are following for a specific therapeutic application, in this case, the COVID or SARS-CoV-2. So I was listening to the discussion today, and one thing that I really enjoyed was to introduce the topic of the target product profile. That's something that many people that start to do drug discovery projects, they know eventually what the target they want to hit. And they know, of course, all the chemistry it is needed to develop, but not many understand what is the concept of target compound profile and target product profile. And I think this pandemic has allowed us to understand exactly what we need to achieve in terms of treat a specific type of disease, patients, what we need to achieve. And I think that was and is one of the information that has helped all the scientific community to understand how to develop a drug. In this case, of course, we're talking about drugs to treat COVID, but the concept itself is we can apply to all therapeutic areas, and I think that's one of the important things that this discussion on this type of drug discovery process, which was really fast. So we had the first drugs coming into the market in less than 2 years. So it's compared with the usual drug discovery process, it is really fast. We know that the approval that took place in the agencies was on a fast track process that helped to make things to come to the market in shorter times. But even though it was clear that it was needed a clear target product profile, that was, without that concept, it would not be possible to have compounds in the market in such a short time. So basically, what I would like to say is that societies, of course, have adapted very quickly to the situation, just by modifying the type of events they were producing. But the important thing is that they have, even so in terms of the pandemic situation, they enabled the scientists to come together and discuss these ideas, and also to discuss in a way that I really enjoy, because in this case, as you know, COVID was a subject of a lot of discussion in media. That was not entirely transparent at certain times, and that is the role also of the scientific societies, is exactly to bring things on track in terms of pure science and allow two scientists to come together, make the questions in an open-mind way, and without the background noise of the media that usually tends to distort the scientific truth. So basically, that's what I want to say, and very loud to congratulate you for this initiative.
Thank you very much, Rui, for sharing your thoughts with us. I agree 100%. I especially enjoy the fact that you mentioned the target product profile. This is something we should be remembered always, not only in the COVID field, of course, especially to us as academics when we want to do drug discoveries. No? So if we don't want to be naive, we should always start from there. So thank you also for highlighting this. So in my original idea, there was that this roundtable should have been divided in a second part, because I wanted also to discuss a little bit about science, the challenge of scientific dissemination and infodemics. Giovanni Maga has recently published a very nice book. Giovanni, I hope that you will mention it, about infodemics and in this COVID era. but we are a bit late, so what time is it? Yes. So we have just less than 20 minutes now, so because I want to close on time. And I don't want that everyone say, you are Italian, so you're not punctual, and so on. So I want to close on time at 5. And so what I would like now to do is just to give you the floor again to you one by one, and maybe you can wrap up and give a kind of close-up if you want to talk, to give your take-home message, or if you want to address the problem of scientific dissemination. Is up to you, but you have just 2-3 minutes just to conclude. Okay, so Giovanni, are you there?
Yes, of course. I enjoyed a lot, all the roundtable and all the excellent presentations.
I'm glad.
And I'll be very quick. So I think one point that very clearly stem from all the presentations that we have heard is that confronting a pandemic, even from a scientific point of view, is a cooperative job. So no single scientific discipline alone can provide solution. We need to merge, we need to talk, we need to share data, because we can build upon the ideas that others can, come up with. So this is a big lesson. We probably knew already, or we felt already it should be like that, but that was the, benchmark for really demonstrating that the only way to confront a worldwide emergency is to collaborate. The second point I would like to make is that it's critical to bring experts together, also to build up a communication strategy to inform not only the citizens, which is obviously essential. WHO made it very clear that if you want to confront an epidemic, or even more a pandemic, you need to engage the public, but also the policymakers, because we have seen some mistakes, hesitation, maybe not properly taken measures or countermeasure, because the scientific advice either was lacking or was not, listened to. So another point that I think is very important is, if we want to fight on one side the infodemics, but on the other side to build up a real strategy for future emergencies like that, we need to bring scientists together to inform policymakers. So that was, my take-home message, and I would like to thank again all the colleagues for the excellent presentation. I learned a lot, and thank you very much for inviting me in this very nice event.
Thank you very much. As I said at the beginning, it has been our privilege because your voice is very important in this field. So we are very thankful for accepting our invitation. And maybe if there will be a third edition, I don't know, I want to think about, but in any case, I will be very glad to count on you even for further events together. Thank you. Armando Gennazzani, we know that he had to leave. So now, Carmen, it's your turn if you want to make some conclusions.
Okay, thank you very much for this nice round table. And in fact, I have not so much to add because I completely agree with my colleague has said. And for the sake of time, I don't want to repeat again because I totally agree. I think that regarding the new drugs that are coming, or I mean, what is the future from my point of view, I think that now we need to try to develop drugs not only for COVID, for SARS-CoV-2, but only we need to think in the future variants and the future pandemic. And I think that maybe look for pan-coronavirus drugs will be maybe the option. And also regarding the target, in view of the problem associated with viral infections, such as the appearance of resistance and so on, maybe a multi-target approach should be the best option, because if I don't want to enter into the discussion of a host-based target as better than viral target and so on, because this is really always a key point, but maybe a multi-target approach, a multi-target strategy will be also an option. And that's all. Thank you.
Thank you, Carmen. I'm particularly glad because I heard the keyword that I love very much, multi-target approach or polypharmacological approach. Yes, I agree with this view, of course, because even in terms of resistance combination, we have already talked about fixed dose combination, which is already on the market. And so I believe that this should be even something that should be followed, and even as a further step, maybe we can even think about single molecules able to hit two targets or the other potential polypharmacological approaches. Thank you very much. And now
So thank you again for this possibility, and I think that event like this is the proof that probably this is the new way to share science and scientific topics, because for the management of COVID-19, but this is a more general discussion, there are many actors involved in the management of the health, of the development of new therapeutical approach. So I think that the lesson learned from the pandemic is that it is essential a cooperation, a scientific cooperation, a transparency from this point of view. And I think that what we have gained in these 2 years is a new method for work, for collaboration, and I think that this can be the point to start an improvement of the scientific communication, and medical work, pharmaceutical work, and so it is essential in net. And I think that in these 2 years we realize we perform the base for a new type of work, and it is. I think that it is the very, very big achievement that we get from these 2 years.
Thank you very much. Carmine, if you want to make your conclusive comments.
Thanks to all the speaker, and so I already showed how during the Escalate for COVID project we shared the results on web page, scientific publication, but each time I speak about this topic, I love to share with the audience this experience. In particular, during the project, we were able to share in a concrete way the results we're generating, for example, by using the virtual reality technology. This allows us, for instance, to share immediately structural information with scientists all over the world at the same times, in the same virtual room, and with scientists from San Diego, in Switzerland, in Germany, in Italy. At the same time, we was able to have a look to the results we obtained during the project. So I think that this is a real way of how the technologies could support in a concrete way the research we are producing with respect to the COVID-19.
Thank you very much, Carmine. Indeed, we here in Urbino were very timely because the student had the opportunity to approach virtual reality this year at the school. So what you are saying, we agree 100%, and we had experience on that even in a very initial way. So Christian, now it's your turn.
I don't know, but I don't have the feeling that the scientific community has problem communicating. The COVID, of course, accelerated the communication, and I think one thing that was, I think, brilliant was that you could download all the papers. Mm. If you're a normal citizen that pays his tax and that wants to read literature and you don't have access to an organization, you can't. You know, and so I think that was fantastic for the general public. But I think that the way we are working nowadays, you need an immediate response. So you do something, yes, no, like, don't like. Even if in our programs now we have the thumb up, like, don't like. No, it's not like, don't like. There's a reasoning, there's thinking behind it, and I think what I would like is that science in general, not only chemistry, but science in general is pushed more at school, more, I don't know. Did you hear, for example, a day where the best achievements, we nearly put a man on Mars with that project, not? When you see the timelines for production of the vaccine, now that has been, you don't hear about that. You hear about Fleming and that they managed to resolve the structure during the Second World War that saved the people. But you don't hear now those stories. That's what you should hear at the news. You should hear about awards sent and having scientific programs, and maybe people would be more interested. If you don't know, you hear what you hear, and if somebody tells you, and he's a lawyer, and tells you, oh, that was crime against humanity, and it's a big lawyer, you think, wow, it's that guy is a lawyer, he knows his job, he knows the law. So the message is very confused. And I think it's very difficult for a normal citizen to decide. Even for us, it's very difficult because we're not specialists of each other's fields, and I think it's very difficult for me. So I imagine somebody who has, A-level scientific knowledge must be even more difficult.
Yeah, thank you very much. First, the applause. Yeah, thank you very much for this further point. As a scientific society, yes, maybe we should even start to disseminate and talk about science even at elementary school. Yes, we are involved also as academics in such programs, but we should do more, yes, and even have a different relationship with new type of communication tools now, and to reach even the young generation, but not just you, because you have already selected your path in science. But yes, this is something that as professors at university we should do, to start to say everyone, at least I would like to say to everyone how rewarding is a career in science, how the life is so full of everything, and so it's hard, but it's joyful. And so yes, this is something that we should even share with everyone, especially for students at school, at the elementary school, so that maybe they can decide to enter program in science. And so thank you also for that. Rui, so the very last comment.
I would say that the major lesson that we can take is in fact that the research that we have seen and the pace that it happened, it is a result. It was only possible with collaboration. So that's collaboration. That's a key word that we need to take, and of course applying this to this disease, but to any other therapeutic application. That's a key information. The other one is about communicating. So that's how to communicate science, but how to communicate properly to specific target audiences. So that's a difficult part. So we at the FMC were trying to reach. So we were advised to not necessarily to reach high schools, but to show that what science can provide beyond the COVID science. I think that's one of the main things that we need to do, is to show the society, and in particular, we are here, I mean, most of the audience is medicinal chemists, so to show that chemistry plays a role in drug discovery, and so drugs are only possible because medicinal chemists can develop drugs. And of course, we all know the story, how fast the discovery of the first anti-COVID drugs was possible, but we need to apply this concept to other therapeutic areas and to show that medicinal chemistry is really important to have new drugs to fill the gap in terms of medical needs.
Thank you. So we are perfectly on time, so now it's my turn to thank all of you for accepting our invitation. I would like to thank Marco, who has hosted us and also supported me for inviting speakers. We have decided together what to say in this round table. I would like to thank all of you because of your patience. It has been a long afternoon, but I believe that you share my feeling that today we have experienced that nothing is so bad, so the pandemic has been very hard. But to me, I don't know, maybe it's just me, but it's the first time where sitting at the table, I have echoed many times words that are the roots of my career, my life in science, collaboration, open science, putting together different disciplines, multidisciplinarity, work as a whole. That to me it's something that we should always consider, as all of you have already said, but not just for COVID. Because I was at the beginning, if you remember, I was a little bit skeptical about talking always about COVID. For example, my main scientific interest is on Alzheimer's disease. Alzheimer's disease are patients who are, these times are even more vulnerable than the others. So, okay, we should find new drugs for COVID. I agree 100%, but we should not forget many other challenges that we are facing as a society. So, but I know that we can count on the efforts of the many actors. In Italy, at least many are sitting here, so this makes me more optimistic. I know maybe the future is not so rosy because Giovanni has explained a lot about the new variants, but I don't know, I feel a little bit relieved after this roundtable, and I truly hope that you share my feeling. And with this, I would like to close this roundtable and thank all of you and also the people connected on the web. And just a technical communication, I would like to make a stop, at least 10 minutes. So let's reconvene again in 10 minutes here. And bye-bye to everyone, and thank you for joining. Bye. Sorry, I should repeat because the microphone was off. So this second part of this afternoon is dedicated to the ESMEC alumni. In English is alumni, but in Latin is alumni award. And I should say that, as we were discussing before, science, the life of the scientist is not just hard work, it is, because it's very hard work every day, but there are also awards, and so it's also important that we are giving this award through a Zoom event open to all the community. And this award is quite new. If I'm not wrong, it is in its second edition. And this year we received quite few number of applications. So the committee had a hard work in deciding who would have been the awardee. And what we have agreed on is that we wanted to recognize a scientist at the early stage of his career, because we were wrong, it's our fault, we didn't publish in the website a kind of regulation, so the applicant should have less than 40 years, but this was our mistake, so we apologize for that. But then the committee decided that this was the target of this kind of award, because this is a school for PhD students, so we wanted to recognize someone who is at the early stage of the career. And so we have decided that the winner for this year is Professor Vittorio Canale. So please join me in congratulating him. So it is my pleasure, Vittorio, to give you this prize. Actually, it is also very nice. Look.
I hope that I will have space in the luggage enough, but still, I will go with the security.
You will find it for sure, because it is so nice. I'm sure that you appreciate. So thank you very much. So we are ready for the official picture. Okay. So, as it is in the tradition of this award, so Vittorio will deliver us a short talk, and I would like just to spend some words by introducing Vittorio. Vittorio was born in Italy, in Caltagirone, in Sicily, but what I believe is a very interesting trait of his career is that then he moved to Poland, so again, we are very fond of contamination, internationalization, and so on, and he has started an academic career over there. He first year his PhD there, and then in 2019, he was appointed assistant professor at the Department of Medicinal Chemistry, where he is currently working. And his scientific activity mainly focuses on the design and synthesis of CNS acting compounds targeting the serotonergic receptor. I also remember nice paper authored by Vittorio, and another point issue that I enjoyed reading, and also the committee reading Vittorio's CV, is that he also is trying to find a new way to synthesize drugs. So he's a synthetic medicinal chemist. He's also interested in developing approaches who are green, sustainable. So this is something that is very appreciable and noble that everyone should consider today. And So is the number of publications according to the CVR 29, but there are also patents and patent application. So, Vittorio, now the stage is yours. We will move on the other side, and we are looking forward to your presentation.
Well, thank you, Maria Laura, for the kind introduction.
Or maybe I can sit here to close the view.
I would like to thank the scientific committee for this opportunity, and I would like to, and it's a big pleasure for me to be here today and to receive this award in presence, and with the possibility to be here again in Urbino, in this beautiful city, and for the opportunity to return to visit even the Ducal Palace with the amazing guide, Madame Lucia, that I thanks for this again, and let's go. That the first time in Urbino was in 2015, and I was at the 4th year of my PhD, and I was presenting results connected to my research, and this is me, yes, presenting, and of course, as you can notice, I have much more hair in the head, and less kilos in my belly, and the poster was awarded and was selected for oral communication. And at the time, we knew about the presentation just the day before, so I was really stressed that I have to present. But for fortune, it was a good organization. We have a concert the day before that helped me in relaxing. And that's at the end of the school, I received these certificates, and thanks to that, I can apply for the award. And that's why I'm here to present you the mainstream of my research. Focus on the development of 5T7 receptor antagonist, and I will show you also the possibility to apply sustainable approach for their synthesis. And the 5T7 receptor is the latest addition to the serotonin family of GPCRs, and is positively coupled to adenylate cyclase and through a GS or G12 protein. And correlation between distribution in certain area of brain and its function suggests an involvement in the learning and memory processes, as well as in the pathomechanism of mood disorders. And in particular, it was found that pharmacological blockade of the receptor by an antagonist provides antidepressant-like properties in animal models. And since my PhD, I was together with Professor Zeider, we were synthesize and develop a class, a group of 5T7 receptor antagonists named as long-chain aryl piperazine biomimetics. And at the end of the project and at the end of the PhD, we synthesized around 120 compounds. All of them were submitted to radioligand binding assays, and the majority of them was very active with a KI value around less than 50 nanomole. And structural activity relationship studies recognized the molecular requirement which guaranteed the high potency. And in particular, we need to introduce a branched alkyl such as isopropyl or the phenyl ring in ortho position of the aryloxy fragment. Then we have to use a tetrahedral H-bond acceptor group, such as sulfonamide, and the tetrahedral conformation of sulfonamide was preferred than the planar one of the amide and the urea. And in particular, we have to keep the distance of 3-methylene spacer between the ionizable nitrogen atom of this alicyclic amine and the sulfonamide. So with this result in hands, we selected lead structure or promising structure that they are characterized by high potency and strong antagonism at the 5T7 receptor and average selectivity over other related serotonin receptor, dopaminergic D2 or alpha-1 adrenergic target. And importantly, they did not bind to off-targets such as HERG or histaminergic H1 or muscarinic M1, so they have low propensity to evoke potential CNS or cardiovascular side effects. And we tested them in vivo, and they exerted antidepressant-like properties in the forced swim test in rodents. And if we compare the potency to the reference compound, we were much potent, and we have similar potency, but the effect occurs at lower doses. And moreover, compound 1361 was also display procognitive activity in novel object recognition task in rats at the doses of 1.3 milligram per kilos. And now I'm moving from the last year from the Department of Medicinal Chemistry to the Department of Organic Chemistry in this new, really nice, well-equipped lab at the Faculty of Pharmacy. And we're still working on this topic. But now we are looking for biased 5-HT7 receptor ligands with potential application in the treatment of depression. However, today I will talk more about the synthetic part of the project, and I will show you the possibility to reduce the use of traditional thermic methods for the synthesis of biological acting compound, and to applying a more sustainable approach as the medicinal mechanochemistry. And the mechanochemistry it could be defined as a process in which the chemical reaction occurs by direct absorption of mechanical energy during the milling of the reagent together. A very old technique is its roots in the Stone Age, when human grinding the food, but the first reaction was described by Theophrastus, and it was the method to achieve the quicksilver, so the mercury, by just grinding the cinnabar using a copper mortar and pestle. For fortune today, we are lucky that we not have to use our muscle to perform this reaction anymore. We have electronic device such as the vibrational ball mill, and in which we have a reaction jar made by different materials such as Teflon or stainless steel. And so the jar oscillated from one side to another, and at very high frequency, and it caused the ball inside the jar grinding the reagent together. Mechanochemistry has a lot of application, especially is known for the application in the drug technology. However, recently, very recently, it could be recognized, it has been recognized as a good approach for as alternative way for the synthesis of organic compound. And the key aspect is the unquestionable advantages that you can have if you use this approach in comparison to the solution one. As you know, in solution we have to solubilize our compound in organic solvent, then we have to heat, we have to cool down, then we have to do a workup, and we have to purify our compound, probably mostly by chromatography purification. Using mechanochemical approach, we are independent of the solubility of the reagent. We just reduce or avoid at all the use of solvent. The reaction are very rapid, from minutes to hours, and most importantly, often we obtain pure compound without any purification step. So we think that we can apply this methodology to the synthesis of one of our lead, 1361, and the synthesis in solution of this compound is consisting in 4 steps, and starting from the alkylation of 2-phenylphenol in a biphasic system. Then we have the hydrophilic substitution with the Boc protected 4-amino piperidine, removal of the Boc, and the obtained primary amine was sulfonylated with the proper aryl sulfonyl chloride to give the final compound. We have 2 problems in this synthesis. One is the first step, that is the limiting one, because we have to use an excess of epichlorohydrin, in order to block the opening of the epoxide ring, so you have the dimer. And another point is that apart from this stage, all the other, to obtain the intermediates of the final compound, we have to purify them by column chromatography. And the third point is that we were searching for a more greener obtaining sulfonamide by avoid the use of dichloromethane. So the first stage of optimization was the reaction of the 2-phenylphenol with the racemic epichlorohydrin, and we start the optimization in a 10 milliliter Teflon jar, and with the ball mill operating at 30 Hertz. And just to make the story short, the potassium carbonate was the best base, just three equivalent of that, and then we increased the kinetic of the reaction by just increase the diameter of the ball from 1 centimeter to 1.5. However, the best reaction condition, best conversion, was achieved when we used just a slight excess of the alkylating agent with a milling time of 140 minutes, and we were able to limit the formation of the dimer just to the 2%. Then we scale up the process, and in this case, we see that we apply the LAG, so the liquid assisted grinding. So it's the possibility to use an organic solvent, just small amount of organic solvent to improve the mixing. And in that case, was the best one. And then to confirm the versatility of the process, we also choose to alkylate different phenol, and we obtain a similar result that in case of the 2-phenol, to substitute orthophenol, phenyl substituted phenol. In the next step, we react just 1-to-1 the alkylating agent with the Bock protected 4-amino piperidine, and to our delight, we were happy to obtain very good yields in this reaction after only 70 minutes of milling. And again, to demonstrate the versatility of the protocol, we also changed the kind of the alicyclic amine, and we obtain a similar result. The difference in the kinetic of the reaction, we just have to prolong the milling. And surprisingly, in case of the 3-amino Bock pyrrolidine, we observe a degradation of the intermediate, but elimination of the hydroxylic group, and this was never observed in solution. So it's something that is just in the milling, during the milling. And finally, we deprotect our amine just by gaseous ACL, and our primary amine was then submitted to sulfonylation reaction, and we were able to eliminate the dichloromethane, and the organic base, usually a 3-ethylamine, and we replace it with the non-toxic potassium carbonate, just one equivalent. And after 1 minute of milling, we have almost 100% of conversion. And then we add to the jar ethyl acetate, and just after washing the organic phase with the potassium hydrogen sulfate aqueous solution, just to remove the unreacted amino function, we obtain compound 3061 with 80% of yields, isolated yields. So we develop all the process was developed using mechanochemistry, and in comparison to solution, we improve the overall yield from 34 to 64%. We reduce the time of the reaction to less than 6 hours, and we simplify the procedure, as all the intermediate and the final compound was obtained just by extraction and not without the need for column chromatography purification. And then we apply a similar protocol to obtain a small library of compound of dually acting alpha-2 and 5-HT7 receptor antagonist. And in this case, also each member of the library was obtained with less than 4 hours of milling, totally solvent-free and without purification on silica gel. And to the best of our knowledge, this represents the first example of a compound library for biological screening, totally synthesized using the mechanochemistry. So just to not stuck to one chemotype, we also develop this method for other scaffold. We use different halogenated isoindolene and as potential 56 receptor antagonist. And we develop a mechanochemical protocol using different halogenated isoindolene and then involving the sulfonamide of different halogenated isoindolene and followed by the nucleophilic substitution with anhydrous piperazine in different position of the sulfonamide of the phenyl ring. For question of time, I could not I could not present all the optimization. However, the sulfonylation of the isoindolene was really smoothly, but this we know. We know before. Just we replace the potassium carbonate with sodium hydroxide, and with a milling time between 5 and 7 minutes. And also we change the material of the jar for Teflon to stainless steel. And in case of the substitution, we observe that the reactivity depends on the position of the fluorine atom at the ring. So the best was the parafluoro, followed by ortho and meta. However, we were able to obtain of the final compound in a very good yield just by using 3 equivalent of pyrazine. 4 hours of milling. We also have to heat the jar at 50 degrees, and we apply LAG using acetonitrile as a lubricant. And then the isolated the compound was isolated just by precipitation with the cold water, then filtration, and we just washing with water excess of piperazine. So just to conclude, I hope that I convince you that mechanochemistry tool for the synthesis of lead structure for preclinical development, as well as for the synthesis of a small library of biologically acting compound in a very sustainable and efficient method and manner. And our developed protocol, in comparison, in contrast to the solution one, not use just use a small amount of organic solvent. We improve the overall yield, and we also not need of the purification step by column chromatography. And we believe that mechanochemistry will be soon a key component in the drug discovery program, both in academic and in the industrial research. And I would like to thank, first of all, Pavel Zajdel, my mentor, that thanks to him also I'm here today. And I would like to thank also the young person from my lab, so Wojtek, Claudia, and Matilda. And I would like to thank also other people that participate in the project. And I would like also to say hello to my wife and to my son and to my to my daughter that I know that they look at from Poland. And of course, all of you for your attention.
Okay, so now with that, that's all finished, the long afternoon. I'm so glad that you finished it by saying hello and greeting your family, okay, because we are not just scientists, but we are human beings, so family is always so important. And so, again, thank you everyone, to the people connected, to the people in Poland, and to all of you for being here. Thank you very much. Looking forward to the next edition.
I may add that if you have some question, I open for the
no,
later.
It's a prize.
And,
it's
yes.
a prize. So when
no, it should be question.
No, no,
No,
no.
not now. After.
Yes, yes, because the price, you know, it's an honor, so. Bye again.
Thank you. Thank you again. Sì. Bello, poi col palazzo.